Some more great Farming Smarter partners to tell you about as we continue with our program. This time, a shout out to the Real Estate Centre, as well as Meadows Canada and Glacier Farm Media. We'll stay with our Farming Smarter family for our next presentation as we check in with Mike Gretzinger with a research outbreak. Mike is the coordinator of small plot research programs and focuses on grant-based projects aimed at producers, agronomists, and other stakeholders to ultimately benefit the public good. He works closely with Dr. Gerbir Dillon on the trials led by Farming Smarter, as well as researchers from other institutions who have an interest in growing things in our neck of the woods. Searching for answers for today's questions in farming.
Okay, welcome to the Research Outbreak. I am Mike Gretzinger, the Research Coordinator at Farming Smarter. I've got some exciting update to give you on 3 of the projects that we're busy working on. The first is going to be our brand new biostimulant trial. And so a special thank you to everybody in that middle picture there. That's our agronomy crew. The 3 Sanders boys, myself and Gerbir, who did basically all the data collection and physical labor on those plots this year. I'm also going to give you an update on the rotational study from Dr. Jan Slasky on the left, and as well as an update on the third year now of our fusarium matrix study with that's Alejandra on the left and Dr. Randy Kuchar. So I'm pretty excited. I've kind of got a theme for this presentation, and that is the systems. And I really want to focus in on the systems approach to these trials and what it means, you know, a cropping system or a rotation and things like that. So we'll get into that. So we've got, you know, a cropping system, a housing system, an operating system. That's where my focus is going to be today. So let's jump right into the biostimulant trial, because this is really new and exciting. And so, you know, when you say the word biostimulant, what does that mean? Well, a biostimulant is just something that stimulates the natural process in the plant. So it might be something that increases nutrient uptake, water uptake. It might help aid in the symbiotic relationship. And so really anything that helps improve the natural system that's already going on. And so that's what we're going to look at. And so there's different kinds of biostimulants. There's fulvic acids, there's humic acids, protein hydrolases, there's microbial inoculants, lots of different categories, but they're all essentially trying to achieve the same end goal. And it's a really similar end goal to kind of the health food side of things. If you need an analogy, like the whey proteins and the exogenous proteins and, you know, herbal teas and all sorts of things, they're really just trying to bring you to that final great level. You know, all the bases are there, the cores are there, but they're really trying to help you, you know, utilize all the energies and nutrients that your body's taking in to maximize what's going on. So why are we studying this and why are we looking at it? Well, it kind of goes back to 2013, and that's when the CFIA made some changes to the Fertilizer Act. And so for better or for worse, some of the things that were happening was that there was a lot of companies who had products available in other countries. There were things that had been proven. They were trying to bring them here, but there's a lot of red tape to get through. And they were trying to bring some innovation into the Canadian marketplace. And for better or for worse, they removed some of the efficacy requirements, and they changed some of the framework for getting products into Canada and Alberta as well. So what that means is that, you know, there was kind of a Wild West. There was a lot of things that came online, but I think that, you know, the things that are tried and true, and anything that, you know, is worth its value is going to stick around. And so, you know, back in 2013, 2014, there was a lot of unknowns, and a lot of producers, you know, they were saying, hey, what about this? What about CO2, boron at 4 to 6 leaf, you know, different seed treatments, foliar products and things like that mix in with herbicide. Like, you know, what do we make of these? Should we be buying them? What should we be doing? So the Canola Council put out a really good study called the Ultimate Canola Challenge, I believe. And it was just looking at, you know, a very reductionist scientific point of view. If you put any particular product in the crop, is it going to give it a boost? And we hope that, you know, if you add, you know, a little bit of this, a little bit of that, you're going to see an increase in yield. But that's kind of where I wanted to get back to the systems side of things, is that I think, you know, this is very reductionist, and it's very useful to say that, you know, this seed treatment didn't improve yield. But that's why we're taking, you know, a slightly different angle on our study. And so, you know, these are the biostimulant trials that we have. These are at Forestburg, Battle River. And then the next shot is going to be our plots in Lethbridge at a little earlier stage. So we're growing canola and peas and wheat. So we're going to grow canola after our wheat and peas after our canola. And the purpose being, or what we're trying to achieve, is every plot that you see here is going to stay that plot for the entirety of the study. So if that's a check plot there, that's going to be a check plot with a different crop on top of it next year. And so we're going to get a look at the entire system. And that's going to give us a lot more power after 3 years than just saying, oh, we just tried one product, a seed treatment, it didn't seem to work. We're going to be able to say, hey, look, we put seed treatment, we did a foliar application, we did another foliar application, we did 3 crops. Like, we're going to have a lot more value in the study itself. So a special thanks actually to everybody who was involved in this study. So ATP, Stoller, Alpine, and Penergetic were really awesome about getting product on a really short deadline this spring. There was a few others who wanted to be and couldn't be involved for a number of reasons. But you know, so this is kind of our treatment list. I'm not going to share all the details. I think there's a little bit of competition between them as to what they're going on. But I mean, if you're working with one of these companies anyways, I'm assuming that the treatments that they're giving us based on, you know, cropping conditions, soil test recommendations, things like that, I'm guessing they're going to be pretty similar to yours, although there might be some variations. It depends really on your unique situation. So that's the other benefit here, is we're really trying to tailor each of these as best we can to our individual sites and things like that. So Farming Smarter, Battle River, and Sarda. This is year 1 of 3 so far. So we're going to rotate all those crops. And this is just a shot of us getting some seed treatments and herbicides, foliars, things like that, ready to go. So we did a plot hop this spring, and this was one of the first cool things that we dug up. So we ran into each of the plot, randomly pulled a couple of plants out, and then grouped them together. And, you know, if you're to wager a guess, you know, there's obviously 4 products. We've got a check. And then we threw an extra check in, and we just threw that in because there was kind of a lot of unknowns, and we weren't sure, you know, if we just thought maybe there might be some comparisons we realized after we get started we want to make, and then we wouldn't have room to do it. So that extra check is called we've got a traditional, which is just NPKS, and then we've got an advanced check, which has like a seed treatment. It's going to get a foliar fungicide, foliar insecticide, whatever things, all the extra things that we think we might put on where we don't want to confound the other treatments. So if I were to show you the 6 there, our 2 are advanced, and our regular check are on the far left side, and then our 4 products are the number 3, 4, 5, and 6 on the right hand side there. So you're seeing some vigor boost, a little bit more robust biomass at our site anyways. And then looking at the canola, similar thing. We've got the 2 checks on the far left there, and then our 4 treatments on the right. So pretty interesting to see like a visual response. I always get excited when we get a good visual response. And then numerically as well, we saw a bit of a trend with a couple products. So what I did here is this is looking at the biomass for peas, canola, and wheat. That's all amalgamated together. And for these next series of charts, I thought it was easier because our biomass tended not to be the exact same time in the exact same range because you're comparing different sites. So I normalized everything to 100%. So that 100% number is where the traditional is, and then everything is going to be relative higher or below that as a percentage basis. So you can see at our Lethbridge site, we had a couple, you know, 5-10% higher. And then strangely, when I added the other sites in, we're not seeing a statistical difference here. There's a couple that look like they're lower, but there's really no difference. That's just the variability. And so we need a couple more site years to really be able to flesh out, you know, is our site, was it just a fluke, or is there something more specific going on here versus other places? And then we can dig into why that is. And looking at yields, so we do have yields for our 3 crops, and I did the same thing. So I went a normalized value at 100%. That's our traditional, the second one from the left. And our average yield was 85 bushels an acre across the 3 sites. And we've got N equals 12, not quite significant. We're kind of 0.014, so there's maybe suggesting something's going on. If I run the line across 100% there, you can see we got one treatment that's standing out that, you know, there might be a yield increase, but it's too early to say specifically that, you know, we'll call that a winner. If I look at the wheat yield, again, you know, those error bars are still pretty big after year 1. And so there's a few kind of above and below, but it's too early to really say if something is particularly working. Now, peas is a little bit of a different story, and there's some crazy things going on with peas. And I heard anecdotally as well that some companies they see their biggest increases in peas. And so it wasn't surprising when I shared some of this with them that, you know, there's a huge jump in some of these. So the Lethbridge and Sarda wasn't as drastic as the Battle River. Battle River had a significant higher amount, higher response to some of these products. So I'd like to figure out what's going on there more. So I actually took the Battle River out just to show, you know, we are still seeing something at our other sites. So it'll be really interesting to see moving forward, you know, how these things work out. So summary for the biostimulant is that, you know, we're seeing some missed wins, we're seeing some missed losses. The peas are really showing the highest, most promising results, and we just need those site years to keep going to see, you know, where this will take us after 2 or 3 years. So I'm going to jump ahead now to the Slasky trial. This is Dr. Jan Slasky's crop matrix, and so we've got a little bit of a matrix theme because I stole some slides from my last presentation. And this is really all about how do we integrate our novel crops into a normal system. You know, staple crops like, you know, the wheat, peas, canola, barley, things like that, they're pretty established, and we kind of know how to work with them. But then we throw a hempen or a quinoa into the mix, or a dry bean or a corn. Where's the best place to put it? And that's what this study is all about, and that's what we're looking at. So we've got, it's a 2-year study. There's a precursor crop and then a following crop, and that's actually what's behind me here. And we're looking at all sorts of things: emergence, biomass, yield, etc. So the main sites of Vegreville, Alberta. We've also got one Farming Smarter, Sarda, as well as Indian Head in Saskatchewan. So the novel crops change depending on the location, but we've got the same staple crops as well as hemp and quinoa for every site that we have. Okay, here we go. So I just wanted to show you a visual comparison. You know, I've got like 20, 30 small plots here on the screen, and these are kind of medium-sized plots, all of these. So that's how much room this study is taking up. And on the right-hand side, that's our precursor crops. So we got 4 reps there of precursor crops. Those are the big rectangles stretching left to right. And then on the right-hand side, that's year 2. So we've already had those big plots, and now we're putting the small plots across. And so that's how we're getting every permutation and combination. So in this, we've actually got 81 plots in each of those small quadrats there. And so there's a lot of data and a lot of plots to look at here. And it makes the analysis quite challenging, because I can't just show you, oh, here's this treatment on this treatment, this treatment. There'd be 81 different things I'd be showing you. And so you kind of have to get creative with how we're going to present things. So just wanted to give you a quick shot. So you see this tractor moving down here. This is me in the next shot, and learned how to drive the tractor. So we're going to cross some of the different stubbles here. We're going across a corn stubble, a barley stubble. We've got some canola and dry bean stubble as well. And, you know, it's really a cool study to be able to get all those different things lined up all at once. And I think we're seeing hemp right here. So it's just really interesting to be able to do this in such a close proximity in a small plot situation. Here's a matrix theme. So our first kind of question that comes up when we're trying to put a novel crop somewhere new, we're trying to grow it for the first time. The very first thing we always say is, okay, what do I grow it on? You know, what should I grow my quinoa on, right? So let's delve in. Okay, well, I got these shots first. So these are some shots of first. We'll go back here. This first one, this is quinoa on quinoa. That's typically not recommended. We've got some quinoa on flax, some quinoa on canola, and some quinoa on barley. And what happens is novel crops are really, really sensitive to different things, different factors. They're just not like a staple crop where you can make a couple mistakes and get away with it. With a novel crop, you're not going to get away with any mistakes. And not even just a mistake, you're not going to get away with, you know, inclement weather or tough growing conditions or insect problems and disease problems. Like, they're just, because you don't have that full agronomy package, all those things factor you so much more. So real challenge here. I don't think these ones were able to be harvested, but I'm going to show you ours here now quickly.
This first plot we have here, this is quinoa on quinoa stubble, and usually a crop on itself is not recommended. You can see there's a little bit of weeds poking through, but otherwise it's actually not as bad as I expected it to be. I think if we went to some of the other reps, they were a little bit weedier with the quinoa and quinoa. So now we've got our corn stubble. This stubble is causing a number of issues compared to some of the other crops. Obviously, we're seeing a height reduction here again, and the weed situation looks okay. We're usually fairly clean in our corn years previous, but I can tell I think this yield's going to be down 10 or maybe 20%. I guess we'll have to wait and see. Moving on, if you're new to growing quinoa, your recommendations are going to be to grow it on a cereal. So this is quinoa on durum. Already I can see and feel, probably on camera, I'm not sure if it picks up, this is a lot taller. So we did some height measurements on all this as well. This quinoa is a lot thicker stand. Even the plants themselves are a lot more robust. So that's probably one of the benefits of not having it on its same crop. As well as the weed control, you can see there's a few sporadic kochias, but this looks really, really good in terms of quinoa fields. Right now, it looks like the winner to me. It's nice and tall. I could feel the crop growing as I drove by compared to everything else. The plants are just really robust and I think nice and uniform. I'm really happy with this, and this is our pea stubble. I'll show you this plot. I don't want to. I really don't even want to try and harvest it, but you can see this is our quinoa on hemp.
Okay, perfect. So that answers the first part of our question. You know, what crop should I grow it on? It's really easy to throw a bunch of precursor crops, cover it all in hemp or quinoa, and then figure out which works the best. But there's two sides. There's a second side. And so given now that we have these novel crops, what do I grow next? What's the best thing to do? You know, can I grow a quinoa on quinoa? Can I grow a hemp on hemp? Or should I move to something else completely different? How do I integrate it, right? This is the integration question. So like I said, all the combinations make it really, really difficult to visualize the data. And so we've got a couple creative ways of doing that. The first is Jan put a report together on last year's findings. And one of the themes that kept coming up over and over and over again was looking at each of the individual charts and sites and metrics that we had, was that anytime we put a crop on its own stubble was a bad thing. And it either usually decrease yield. It also sometimes increase volunteers, weed pressure, things like that. There was a lot of other unintended consequences because of some of the different some of the different factors for novel crops. For instance, lack of broadleaf controls in some of our hemp and quinoa makes it so that higher weed pressure is going to be an instance the following year. So a lot of the metrics showed that crops performed poorest when they're on their own stubble. We kind of already knew that, but this is just an illustration of what's going on. And so all I did here was I ranked all the sites, Lethbridge, Vegreville, Sarda, and Indian Head, 1 being the best and 9 being the worst. And this is the rank when we put it on its own stubble. So if we put a crop on its own stubble, is it in the middle of the pack? I mean, if it didn't matter, we'd expect it to be pretty broad. It, probably be a normal type distribution here, but this is skewed entirely to the bottom of the scale, which is saying that, we have basically our lowest yield every time we put a crop on itself. And we kind of knew that. This doesn't mean that all these were significant. I just went numerically, but there were a lot that were significant. Sometimes the significance shifted depending on what part of the graph. So it's just a way to visualize and just show, like, don't plant something on its own stubble. And then if you look at the hemp, kind of interesting, I've got 3 hemps up at the top where hemp did the best. And I think there's a couple of factors for that. One, our hemp yields weren't great that first year. We had a snowfall that came in, if you remember, September 30th. I don't know if all of us got harvested before that happened. So there's an asterisk on all those hemps there, because I think our yields were only about half of probably what commercial fields were, and the data wasn't significant as well. So I guess we'll kind of leave that for what it is for now. So I thought a good analogy for kind of thinking about our rotation is kind of the blood donor system. So you've got donor, i.e. donor crops, and you've got receiver crops. And so, you can think of some crops, like we think of like peas and pulses and lentils, things like that. We think of that kind of pulse area as a really good donor crop. Like, we'd love to be able to grow every crop on a dry bean around southern Alberta, but that's just not possible. That's only one piece of your rotation. And so you have to have receiver crops. You have to have crops that are good at being seeded on any stubble. And so that's kind of what I'm thinking is a good way to think about this here. So this is a shot of one of our reps. And you may have seen this before. I just took all the yield data and piled it on. And so what that's going to show you is that's going to show you which crops perform, better than others. And for a given crop, it's going to show you which was the best stubble. So the stubbles there on the left, if you were to look at the wheat as a year 2 crop and go up until you hit dry beans, that's going to be your best yield, 52.75 kgs per hectare. But that doesn't tell you what you should do after you grow wheat on dry beans. And it doesn't tell you, what you should grow before the dry beans. And so a better way to do this is to kind of give this an index and to normalize each of these crops as a percent of themselves, as a percent of the max. And so that's what I did here. And so that way we can compare, each of the crops to one another in a relative kind of sense. And so I've updated that from what I did before. And so I just wanted to kind of show you with that donor / receiver type analogy that, our cereals, we kind of know this already, especially in research. We tend to almost universally like put things on wheat stubble because we know it's a pretty good receiver or donor type crop. So durum, wheat, barley, they're all really good as stubbles. Likewise, they're kind of like the O negative here. They're really good as crops as well on any stubble. We can put a wheat on pretty much any stubble and it's going to perform relatively well. So when we're thinking about how to integrate our novel crops, that really helps us figure out where the best place to put them are. So if you look at our novel crops, for example, like hemp, quinoa, corn here, there's going to be some spots where they do just fine. But then there's going to be some problem areas, and that's kind of where we want to focus. Like I said, crops on themselves do poorest. Quinoa and quinoa has got like half the index that quinoa and wheat has. And so what that means is that, you're going to do far better off putting quinoa on something other than itself, and especially problematic for quinoa. Wheat on wheat, you can probably get away on that with that a little bit better than you would with something with quinoa or something like hemp. And so that's where this index comes in, is it kind of shows you the relative differences. And there's going to be crops that are really good at buffering. You can get away with just about anything, but novel crops are kind of special that way. And so I thought I'd propose Trevor and Lewis to a challenge here, and they don't know this, so they actually didn't get any say in what they decided, but I decided for them what they would be. So I challenged them to a rotation arrangement to see who could come up with the best crop rotation. So I got 3 crop rotations. I've got wheat, peas, and canola. Or sorry, we each have a crop rotation, and we've got the same crop rotation. We're all growing wheat and peas and wheat and canola. Like that's our rotation, 4-year rotation. And our challenge is to input hemp and quinoa into that rotation and do the best job possible. So Trevor's like, I don't really want to participate. I'd rather just roll my barley and spray some other crops. I'm just going to tack the hemp and quinoa onto the end. Doesn't matter. Good enough. Lewis been paying attention. He saw my presentation last year and he said, no, okay. Hemp, I'm going to put that on wheat. Quinoa, I'm going to put that on wheat. That's how we always grow them for our research stuff. I got it figured out. I know what's going on. And me, though, I've been doing the analysis and playing with all the numbers. So I'm going, okay, yeah, I'm going to keep my wheat on quinoa on the wheat. I'm going to keep the canola after the quinoa. We saw that do good. But I'm actually going to play around with this hemp, and I'm going to go through each one of these spots and see if I can find the spot where I think it does the best in the index. Now, I haven't analyzed this for statistical significance, but it's just to illustrate kind of the power of what this matrix can do. And I thought it was just kind of fun to show. So here's Trevor's economic analysis. So I've got his rotation across the top, and then underneath that, the line underneath is the index. And so I've given each of the index from that previous chart. And then I've added the yield underneath that. So that's his yield in kgs per hectare. I've got a unit price, so bushels per acre, pounds or dollars per pound, something like that for each of the crops. And then I've come up with a gross revenue per section. So I'm going to pretend we're each growing a section of each of these per year. That's 3,200 acres. That's a pretty fair size farm. And my picture's in my way, so I can't quite read what that is. But I think Trevor was about 1.6 million in gross revenue. So looking at Lewis's now, you can see he did a little better job fitting that hemp and quinoa in. And so he's got a better index. What that means is he had higher yields in those plots, and he's going to have a better gross revenue. So he's at 1.75. Now, like I said, I kind of cheated. I played around until I could get my hemp in the best possible space, and I'm at 5.45. So I whooped them both. I'm at 1.8. That's almost 100,000 more per year. But I haven't done any real work. All I've done is change where my crops are going in the rotation, and that's pretty amazing to see that something so small could have that big of a potential impact. And so I think that's really the power of this. And I hope that, once we can, at the end of this fall, we can amalgamate all the site years, all the locations, bring in, a lot more complex layers, and, kind of glean out some of the data where we say, here's got a really high index, but it was bad for all these reasons, so you'd never do it. For example, quinoa and canola might look like it has a high index, but a lot of it's doggage, a lot of it's volunteer canola, which is impossible to clean out of quinoa. So you got to kind of take this with a grain of salt, but this is where this is going, and it's pretty exciting and cool to see. So really what I'm getting at is that, we're looking at how do you rob Peter to pay Paul? Sometimes we're so focused on getting maximum yield all the time, everywhere, that we forget, sometimes you're better off maybe taking the 98, 99% and then getting better overall. You gotta look at that big picture, and it's really cool study for that. So, excuse me. To summarize, I mean, we know novel crops like hemp and quinoa, they cause big problems, not because they're weedy this year, but because they're weedy the year after. And that's why some of the novel crops perform so poorly on other novel crops, is because you don't have your weed control in a row. And the other thing is choose widely, because the novel crops, they show the agronomy gaps much more strongly than, say, your staple crops. The staple crops are really good universal donor receivers. The novel crops, not so much. So that brings us to number 3, and number 3 in our framework is Dr. Randy Kuchar's rotational study, and this is really looking at kind of the same matrix design and a lot of the same overlapping crops. But what we're going to do now is we're going to layer on an entire different aspect. And so the focus of this study is Fusarium head blight. And we're going to layer on a third year of crop rotation, which is just entirely durum, which is, you know, really susceptible to Fusarium. And so there's a bunch more sites participating in this one. There's between 6 and 9 crops within the rotation, depending on the regional suitability and things like that. And so we just kind of hit our third year where we've done this now. This is the third year. So we just did durum across everything. And so we've got some kind of interesting things to show you. So Fusarium head blight is really, you know, it's been an issue for a while, and we have to figure out a way to, you know, they've taken it off the Pest Act, and, you know, that's because it's here, and so we have to figure out a way to manage it now. And so there's a number of different methods for doing that. And so you've got cultural controls, so that's things like the irrigation scheduling, Lewis's Spornado type project. You've got genetic controls, so there are some new genes coming that can hopefully do something, and then you've got herbicide. But these are all things that potentially costly in some regards, and also they're very finicky. They're very specific timing for the irrigation scheduling and your herbicide application, and they're not entirely 100% effective. So what is another way that we can control things? It's almost basically free, is our rotation. And so that's where this system comes in, and it's really cool to see the data on this. So this is Alejandra's poster. She's working on her master's at the University of Saskatchewan right now. And she shared this with me last week, and there's a couple really cool things I just wanted to show you. So the first is that, like I said, it's really hard to analyze the matrix with 81 combinations. Never mind the fact that now we've added a whole layer of durum on top. So in order to kind of segment each possible, I guess to come up with a category for each thing, is she's come up with this diversity criteria. So we're going to look at the treatments as, are they diverse or not diverse? So everything has a cereal, but some of them also have either a pulse or an oil seed, or they have both a pulse and an oil seed. So if they have just maybe a pulse or an oil seed in the 3-year rotation, we're going to say they're less diverse. That's a B in the middle. If they're just all cereals, there's no diversity there. And then if they have a cereal pulse and an oil seed, that's maximum diversity. So we're happy with that. And that's kind of the criteria for analyzing whether or not these systems are working without having to look at every combination of specific crops, so to speak. So this is the percent pathogen contamination. And so, you know, 1% approximately corresponds to about 1 ppm at the elevator, and, you know, 1 to 2 ppm, depending on your class, that's where you're going to be downgraded. And so that's pretty significant and pretty important level to be below. And so if you look at what she's found here, is that this is the criteria for Saskatoon. This is the durum kernels after the year 3. And on the left there, C is our low diversity. That's just all cereals. And you can see we're hitting up 1, maybe 1.5% pathogen contamination. Then as you increase the diversity, it goes down. And then as you increase it even more, we're having a cereal pulse and an oil seed in the rotation, we're down below 1%, which means you might not end up with that downgrade from the FDK. And so she's got Fusarium species, and then kind of broken into some of the individual species depending on how intense they were. So hoping to get this data together for all the sites relatively soon, and then it'll be really interesting to see how effective our rotations were at reducing Fusarium across the other sites as well. But pretty promising and pretty interesting result here. So the Fusarium species were affected by the crop diversity as well as weather conditions, and that's just another layer we're going to add on afterwards. So she's quite interested in the Fusarium aspect. I'm quite interested in the yield aspect. So I just thought I'd throw the yield data on top of this here. There's a couple interesting things coming through. The first thing you might notice is that really big dip That's the durum. Durum on durum. We already said don't grow crops on themselves, and it showed up in particularly strong in this trial here. And I think there could be some issues with weeds, wild oat resistance, and things like that is a big problem. So it's not just the fusarium in our case, but also the FDK. So that's really interesting here. I just wanted to point that out. Now, the other thing was I thought it'd be a challenge, again, to have that whole matrix. So I really liked how she broke things up into cereals, pulse, oilseed. I just added an extra category so that I could throw in all our novel crops. And so I said, if we have a cereal plus any two of those other three categories, that's pretty diverse. That's as diverse as we can be in a 3-year system. So we've got diverse. If we just have one of the other ones, it's less diverse. And if we still, again, no cereals or just all cereals, there's no diversity there. And so now I wanted to look at the yield data through that context as rather than crop specific. And what I found was awesome. So what I found was really similar to what she saw, was that, we're getting a double whammy here. So we're getting reduced fusarium by increasing diversity. And we're also getting increased yield. So if you look at the low diversity, we're hitting 81 bushels an acre with a pretty tight error. And if you go up to medium and high diversity, we're getting about 84 bushels per acre. That's 3 bushels an acre. That's pretty awesome from doing nothing more than having the right rotation or having the right, total diversity within your rotation. And so that's a big win here. And I don't have the stats on this to show you yet, but I think that's just kind of a cool thing to add into this study. So, if I was to summarize all 3 of these trials, I'd say, that when based on that system, rotation, it decreases fusarium. I think it's going to increase your yield. And tune into Trevor's talk and see what rotation does to soil health as well. So thanks very much for listening, and we'll see you soon.